So I'm Jonathan Pritchard, and welcome to the Keynote talks for this year's Biology of Genomes. So it's a real pleasure and honor to introduce Andy Clark for one of this year's keynotes. Andy got his undergraduate training at Brown University before he went on to Stanford to receive his PhD with Mark Feldman. At Stanford, he worked on problems in theoretical population genetics, and he also became interested in fly genetics. After a couple of short postdocs, he took a faculty position at Penn State and then moved to Cornell in 2002, where he's been ever since. Andy's won a lot of awards. He's a member of the National Academy of Sciences. When I think of Andy, I really think of a couple of things. First is that he's consistently thought about new problems in really creative ways that, that within a few years have become central problems in a lot of population genetics and evolutionary biology. And a second contribution that I'd like to talk about as well is the way in which he's really mentored many people in the field. And so so I'd like to just show you a couple of slides about this. So some of you may know that I was an undergraduate at Penn State at the time when Andy was there. And so this is Andy in 1993. So this is about the time that I was there. He's skiing, as you can see. And so the first population genetics course that I ever took was in 1991. And this is part of the syllabus from Andy. So I was 19 at the time, and this class just had a really great impression on me. Made a great impression on me. So I'm still working on the same stuff 25 years later, and so the impact that Andy had was terrific. And so just in case you're interested, here's the syllabus. So we've got 2 lectures on Hardy-Weinberg. So guys, Hardy-Weinberg ain't what it used to be. So we've got drift, neutral theory, models of selection, and so on. So that's just the first half of the course. And one thing I thought that so I pulled my notes out of a box to prepare for this, and so one thing I thought that I would share with you are the sample paper topics that Andy suggested. And so this is 1991, so this is 26 years ago, and I just think that they're incredibly pertinent even today. So here's a list of the topics that he suggested. So we've got inference of natural selection from DNA sequences. So we've got a bunch of that in the meeting this year. Inference of recombination from DNA sequences. There's a lot of that. Evolution of recombination modifiers. What else have we got? We've got implications of selfing, evolution of transposable elements, genetics of speciation, host pathogen coevolution. I just think that this illustrates Andy's ability to see really important problems, and of course these problems that are still in many cases relevant 25 years later. So just to end, I'll show you one question from the final exam. And you can think about this over the wine and cheese. A major news story describes the results of a large study of identical twins. It reported that the heritability of warts collected from twin samples was an astounding 0.9. And so if you want to consider heritability of warts more, maybe Andy can fill you in on the details of that. So, okay, without further ado.
Well, I guess I'm obligated to start with a soggy kind of comment, which is, one of the highlights of the meeting for me was actually meeting my first academic great-granddaughter who's at this meeting. I think that's kind of cool. Anyway, so if we could start with the first slide, please. And I don't feel that old. So, okay, so junk evolution. This is an image of a meiotic prep of Drosophila melanogaster in fish. In situ hybridization. What we're looking at is the red is euchromatin, the blue is this other stuff, highly repetitive DNA. And I'm going to be talking about a subject that composes at least half of the genomes of most of the organisms we're talking about, including flies and us. And despite that fact, only 3 of the 350-some abstracts even mention the word heterochromatin. So I'm going to try to bring you up to speed a little bit. So this topic kind of burst onto the scene with Susomu Ono in 1972, first saying that, okay, it must be junk because we don't know what it does. It's highly repetitive DNA that seems to be in the genome, but we don't know how it's structured or anything at that point. We just knew that there was a lot of it. And then in 1980, both Doolittle, Sapienza, Orgel, and Crick came out with these pair of papers that commented on the fact that because of the way that this DNA was replicated by slip strand kind of processes that made it rapidly expand, it must be selfish. It's not evolving under normal rules. It's evolving under this process of that's sort of molecularly based. And Gabby Dover jumped on this and wrote dozens of papers and at least one book that he called Molecular Drive and used it to explain just about everything and essentially killed the field for at least 20 years. And you notice I don't mention any of the particular papers because some very respectable editors are in the audience. So. All right. Of course, we now know, fast forward to today, that there are a lot of very interesting features of heterochromatin. I think the main reason that we don't see more work done on it is so difficult to make progress with understanding it. It is very rapidly evolving. So the first observation here came from the fact that Doug Brutlag, back in even, I think, starting in the 60s, certainly in the 70s, was doing those cesium chloride gradients, which you all know how to do now. And with cesium chloride gradients in Drosophila, there's a portion of the DNA is AT-rich. So this heterochromatin material that we're talking about is like millions of copies of AATAG, very simple K-mer type repeats. Very, very high redundancy and tend to be AT-rich. So in a cesium chloride gradient, there's enough of the same buoyant density that it forms these sharp satellite bands, hence the term satellite DNA, which you've often seen. So Alan Lowe and Doug Brutlag then got together and did this with multiple species of flies and saw that and sequenced the material from those bands and noticed there's almost a complete turnover of heterochromatin between even melanogaster and simulants. So it's fast evolving. And some of the various functions, I have to speed up here, centromere function, of course, there are centromeric histones, and those associate with specific repeats in the centromeric heterochromatin. And those proteins are fast evolving, and also the DNA is very, very fast evolving. So Steve Henikoff, Harmeet Malik, many others have worked on that problem. This process can go awry, as Nitin Fadnis told us about. And you can actually have selfish-like processes where there's a tendency of particular sequences to end up in the egg as opposed to polar body, resulting in a form of meiotic drive. In addition, the telomere material also is showing very rapid evolution. And the proteins that associate with telomeres, this is recent work from, where am I? Oh, okay, so we'll get to that in a second. But the initial observation that telomeres are associated with proteins and telomeres have to undergo a process to be maintained was, of course, established by Grider and Blackburn. And the material there is also fast evolving. We'll get to that in a couple slides. These heterochromatin regions serve as graveyards for transposable elements. They tend to have low recombination, so the elements land in there and die there. And that has evolutionary consequences. And we've heard a couple of examples where divergence in these processes can result in actual interspecific hybrid dysfunction and hence speciation. So how do we study these? In addition to doing cesium chloride gradients and then sequencing fragments from those, fluorescence in situ hybridization has been very useful. And as recently as just a few years ago, Evan Eichler's lab was quite interested in centromeric heterochromatin evolution. Comparing chimpanzee and gorilla, you study these pictures and you can see they too are showing extensive rearrangements, changes in the structure and organization of these centromeric heterochromatin regions. What we'd of course all like is to have telomere to telomere sequence assembled, and this is only going to come in a number of years from now, but through some kind of process involving very long reads, and that's necessitated by the fact that these very high redundancy materials are extremely difficult to assemble unless you have high reads. So pushing the read length sort of technologies like with PacBio or 10X some individuals have made some very good progress with this. And I think some of the nicest work I've seen is from Amanda Laracuente's group looking at the assemblies of the PacBio assemblies of the Drosophila melanogaster genome, and others now as well. This responder element that, again, that Fadness talked about briefly, involved in the segregation distortion process, has a very characteristic structure where it starts to get contaminated with transposable elements towards the end, but it's many, many repeats spanning 300 kilobase pairs. The 1.688 repeat was one of those that Brutlag talked about, referring to the buoyant density of the repeat, and it also has this fairly pure center, and then it degrades as you go out. So there's even some fine structure at this scale of these particular repeats. Now, Harmeet Malik was the one who recognized that, wow, to make progress on this stuff, because the DNA is such a bear to work with, why don't we just focus on the proteins first, because we know about proteins, a very, very sensible place to get started. And so focusing first on HP1, heterochromatin protein 1, and looking at polymorphism within species and divergence across different species, what Malik and colleagues observed was that, in fact, all these proteins are very fast evolving. They have excess non-synonymous to synonymous substitution, and they're showing all the signatures of positive selection, like they're being driven along by some kind of chase-away process. Now, to this day, we still don't know exactly how that works. It could be that there's a molecular process driving the heterochromatin repeats, the DNA, to evolve rapidly and just kind of turn over by a molecular process, and the proteins are doing a chase away to follow those molecules, and that the fact that they're so different between species isn't a particular adaptive consequence other than the fact that it's making them diverge quickly, and hence resulting potentially in these kinds of Dobzhansky-Muller interactions when they hybridize. But the fact is that it led to a number of key insights about the structure and evolution of centromeres and centromeric proteins, as well as the repeats that associate with them. So this is what I started to stumble into, which is that the telomeres, this was just recently published work from Mia Levine's group, who was a postdoc with Harmeet for a bit. She assembled a collection of proteins that we know are associated with telomeres and looked at those very carefully in that same comparative kind of context. And they too, many of them, show an excess non-synonymous to synonymous variation, a lot of blinking on and off going from one species to another, and sometimes confusing perology. And so they too are showing this same kind of signature of fast evolution, perhaps also driven by the underlying DNA evolving under a strictly molecular process. The other key aspect about heterochromatin, of course, is the fact that we call it heterochromatin because it is a chromatin state. Namely, you can't talk about this without talking about the fact that there are other proteins, there are histone modifications, there are aspects of methylation of the DNA. The sort of whole epigenetic landscape is different between what we classically call heterochromatin versus euchromatin. And the key signatures of heterochromatin, besides the binding of a couple of key proteins, namely HP1 and this one SUVAR39, which happens to be a H3K9 methyltransferase. So this histone modifier does that histone modification, which is now one of the classic signatures of heterochromatin with respect to the histone modifications. This results in a very dense compacted state where, of course, it becomes then gene poor, low recombination rate, and the whole 9 yards. And so that's what makes it largely become a graveyard for transposable elements as well. Digging just a little bit more into this chromatin state, we can look at the chrome HMM sort of picture of the world, and it would be state number 7 on this table, where again you can see high density of H3K9 di and trimethyl in presence of HP1 and that SUVAR39 protein. And so none of the other states particularly have those characteristics, and it's very easy to spot when part of the genome is, strictly speaking, heterochromatic. All right. So we took the challenge and said, well, we're just tired of this giving up on the DNA. Can't we do something with it? And you realize, well, what's the problem with it? AATAG a million times. If you have a bunch of reads that look like that, there's no way ever you're going to be able to assemble it. So the response there is, so give up. Don't assemble it. See what you can do without even assembling the material. And so the trick is actually very, very simple. It's just to take these sequences, one read at a time, and tally their features. And the features that you do are just make a hash table of all K-mers, all words from length 2 to 20, and you end up with then counts of AATAG. This read has 5, this one has none, this one has none, this one has 10, and so forth. And you end up kind of with a fingerprint, a composition based on counts of reads in each of those different classes. And it ends up looking like this, that fingerprint I described. This is the work of Kevin Way as part of his PhD thesis when he was co-advised by Dan Barbash and me. And so you rip through, there are many, 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 of course, words between 2 and 20 letters long, but it's only a small subset where you see these very high abundances of them. And this is just sort of an indication. So I've mentioned AAGAG, the GAGA factor binds to this particular kind of DNA, and there's a lot of it in the fly genome. And flies particularly like 5 mers, some other species really like 7 mers, and so forth. So you get kind of a picture. And it has a fair number of degrees of freedom, but it's not, it doesn't tell you anything about position. There's some things that you'd like to do a little better than that, but it's a start at giving an image of what is the heterochromatin like in this particular sample. Just do want to point out that, in fact, it's highly repeatable if you do the library construction in exactly the same way. So what you'd like to do is to get a tool that just any genome sequence out there, any bit of DNA that you could, or bit of sequence that you could pull off the web, you could run this tool on. And in fact, it's fairly sensitive to the chemistry, to the library prep method, and so forth. But if you do the experiment at the same time, make the libraries the same time, it's highly repeatable. So these are biological replicates, just from the DNA level, taking it all the way through, and then collecting the sequence and analyzing these K-mer frequencies in those libraries, you can get good results. And so everything I'm going to be talking about is that kind of comparison. And it's a sort of a caveat to just run out there and apply it to everything. So we had been working with a set of Drosophila lines that are fairly geographically dispersed. Let's say there are 84 lines, roughly 18 or so lines from each of these 5 different populations. And this project started quite a while ago, so we only sequenced them to 12x, kind of embarrassing at this point, but it's enough for all the things that we want to do still. We got quite good, they're highly inbred, so it's quite good, easy to get quite good SNP genotype scores on them. And when we run this K-mer analysis on those, each of these is a line, and it's like those microbiome images, if you will. So each different color is a different K-mer repeat. And the 5 most common K-mers are indicated with the colors, and the rest are sort of in gray. But you can see this yellow bar is a particular 10-mer that's quite frequent in flies. And you can see these are all female sequences, so there's no Y chromosome in this yet. And so they vary by a factor of 5 or 6 in abundance of many of these K-mers. So they're really different from each other. The other striking thing was that from one population to another, those 5 kind of continental populations, they look like they somewhat overlap in this principal components analysis plot, but you actually, if you build a classifier, you can tell the population of origin of a single fly just from its heterochromatin sequences. There's some particular things that make this a little bit easy. Not many, but several of the K-mers are essentially absent in some populations and relatively abundant in others. So this process of taking off and expanding in some populations and not others seems to be just ongoing at a fairly short time scale. And if you do fluorescence in situ, you can actually see that these things do tend to associate with the centromeres for the most part in flies. Okay, some questions. What are we going to do with this tool now that we have it? We can start to ask, what are the patterns of polymorphism and divergence in different species and populations of flies? What, if any, are the phenotypic consequences of that kind of variation? What are the evolutionary forces that might be maintaining that variation in heterochromatin? And that means, in part, understanding something about the mutational process. So can we actually quantify the rate at which these things are diverging? And so it turns out the Y chromosome is a terrific tool for this, because the Y is mostly heterochromatin in many organisms. In Drosophila melanogaster, the Y is 40 megabase pairs, and it's probably 90% heterochromatin. And it's male-restricted, non-recombining, all this stuff, But it's huge and mostly heterochromatin. That means you can kind of titrate the amount of heterochromatin in a genome by crossing in extra Y chromosomes, or by now if you just compare across lines, you can actually compare what are the effects of having different amounts and structures of heterochromatin as you vary from one Y chromosome to another. So this means making Y chromosome replacement lines, and this is dead easy. There's a line that the Bloomington Stock Center has that has markers on all for the major chromosomes in flies, and with just 2 generations, if you set it up a big enough cross, 1 out of 32 flies will be of this sort that's totally replaced. So its genome is entirely the same as this initial line, except it has this new Y chromosome now. So you do that across all 84 lines. All the lines are genetically identical in their entire genome except for the Y chromosome. How do you know that? Well, you sequence their genomes, and you sequence the RNA from them as well to see if there's any effect of that particular construct on gene structure or gene expression. So the first thing we do is we run the K-mer analysis. So I should mention, we did do the whole genome sequencing to only 1x depth. 1x depth tends to be plenty for this K-mer analysis because there's so many of those repeats. At 1x, if you go beyond that, it's totally saturated. It's stable. The estimates that you get beyond 1x are going to be the same as if you do 100x. So, with the rest of the genome, we also compared, and there were about 10 SNPs across the rest of the genome, which is a bit fewer than you might expect by mutation, but anyway, that's what we saw. And so we were pretty happy that the replacement process, as simple as that was, didn't get screwed up, and we end up with lines that are very cleanly Y chromosome different only. And so the differences then in the K-mer abundances that we're seeing are all on the Y chromosome, and they're quite extensive, and Kevin put this story together, it's coming out. And in addition, because we had the sequence, we could look at transposable elements. The only place that transposable elements could differ in those lines are on the Y chromosome. If we zoom in on the top here, just to see this a little bit easier, these are a bunch of these different males from Ithaca, Beijing, and so forth. Again, these color codes showing different relative abundances of different elements. And the females are here. So again, you can see males have way, way more total heterochromatin than females. It's, a factor of 3 and 4 and even 5 times more heterochromatin than females do. And the distribution of transposable elements is likewise somewhat different because of the mostly dead ones that have arrived on the Y chromosome. If we kind of look at these K-mers by just sort of ranking them by K-mer now, these are sort of in rank order relative of the male-female relative frequency. There are a whole bunch that are male-specific and a whole bunch that differ in relative frequency between males and females. So the red dots are females and the bars there are males. And some of them, like this is that very, very common 10-mer, tends to avoid the X and the Y chromosome. So any repeat that's on centromeres of the other autosomes is going to be the same in males and females, and that's what we see. That was nice. That was known. AATAC here is one that had been known to be Y-linked from Brutlag's work, and in fact we have a much greater abundance on the Y chromosome than we see in females. So there are a few positive internal controls here that we knew about already, and we have a very good complete picture now of that difference in heterochromatin. The other thing that's kind of striking, and we're having trouble making a whole lot of sense of this, is that they're highly correlated. So many of the repeats will be organized in clusters that are highly positively correlated with each other. So that might happen if there's like block duplications of chunks of several elements that are expanding and contracting together. And this is where we're going to need to know some of that information about the higher order structure of it to really, really understand that. This is just zooming in on some of these clusters. So in fact, this collection of transposable elements, DM88 is a copia-like element, and these particular repeats, those are all positively correlated with each other. And then, no, I'm sorry, those are negatively correlated with each other, whereas these other sets, have I got this right? Goes one way or the other. So here we go. McClintock. Yeah. That's too hard to do. Anyway, they're one way or the other. Oh, here we go. Red's negative. So these are the ones that are positively correlated. These are very negatively correlated. And so there are blocks that go up when another one goes down. And why is that happening? The simplest explanation is, in part, it might be this block-like structure. Some of them are very clearly just because of population, that wholesale gains and losses of elements in particular populations. And the huge outlier on the Y chromosome is Beijing. The Beijing Y chromosome of melanogaster is truly odd with respect to these repeats. Some of these population-specific ones show patterns the other way, where it seems to be like only present and expanding in one population, not the other. Of course, these zeros may not be absolute zero. It's modulo the degree of sampling that we've done, but relative to this, it's much, much less frequent than you see here, and so it's essentially population-specific. Okay, now let's move to that question of phenotypic effect. We've got some picture of, okay, yeah, they're variables, so what? Phenotypic effect is something that has been thought about for a long time, and it's was first discovered really by H.J. Muller, looking at a particularly interesting inversion on the X chromosome of melanogaster. And it's this particular inversion that puts the white locus, which is fairly close to the tip of the X, close to this heterochromatin, the pericentromeric heterochromatin. And when genes are put close to heterochromatin, there's this so-called spreading effect, where there's some degree of heterochromatinization of the local chromatin of that gene when it's in the proximity here. And the consequence of this is to give this phenotype, this variegating phenotype, this phenomenon we call position effect variegation. And that's a particularly intriguing thing. It's showing that there's some compression of the chromatin state, making it more heterochromatic, which would shut off the white locus, giving these white patches, but that in some cells, the chromatin state is a bit more open, giving expression of the white locus and hence the red spots. So there's this kind of cellular memory. There's a snapshot taken of the chromatin state at a particular time early in development, and then the cell lineages expand and you get these patches in the eye. That's what gives that beautiful phenotype. And of course, as soon as this was seen, what happened was individuals did screens for suppressors and enhancers of that phenomenon. These are known as SUVARs and EVARs. And a suppressor variegation is one that prevents that chromatin compaction from occurring and hence retains the open state, so you get expression of the white locus and hence the red eye. With this gene SUVAR39, which is one of the suppressors of variegation that was successfully mapped, it turns out, depending on the dosage of SUVAR39, if you have one copy, you see some degree of suppression. If you have no copies altogether, that fly is actually viable. You also get a reddish eye, but overexpressing it gives this degree of compaction that we're seeing here. All right, so that's an interesting phenotype. Can we use it to sort of as a proxy for measuring chromatin state? And the basic question here is, does the Y chromosome, by virtue of it having such different amounts of heterochromatin, impact that particular phenotype? And we can do this by simply crossing our Y replacement line to a line that has this white modeled for. And there are a couple classes of offspring that we get out depending on the second chromosome, which I'm not going to go into that detail. I just thought this slide at least showed this very simple cross. The F1 hybrids is what we're going to be scoring. And the F1s then are going to ask, do the different Y chromosomes impact chromatin state of the X chromosome right around the white modeled 4 locus? So it's a very targeted, are we affecting chromatin state at white modeled 4? And I wouldn't be showing you this if it weren't for this very striking result, which is it goes from almost completely wild type to completely white eye. So there's a massive effect of a transacting effect of the Y chromosome on chromatin state of the X. And it shows some degree of population differentiation because the Y chromosome K-mers did, so that's not too surprising. It's somewhat correlated with total amount of heterochromatin on the Y, but as you'll see in a second, it's much more tightly correlated with specific elements on the Y chromosomes. I should mention Bernardo Lemos, several other people have observed this. So this paper in 2008 was the very first one that showed some aspect of the Y chromosome impacting expression elsewhere in the genome. And this phenomenon actually has been known for years. I think the first paper I could find that described it was actually Hennekoff described it in great detail. Zuckerkandl talked about this aspect of the kind of transacting influence of parts of the genome on other parts of the genome. Now that we know about epigenetics, it's actually quite sensible. The sync hypothesis says that more doses of the Y chromosome de-represses the rest of the genome. And it does that by virtue of kind of soaking up the heterochromatin proteins, so HP1, SUVAR39, so that there's less available elsewhere in the genome. It's a very dumb-sounding idea, so simple, and yet it's highly predictive of a lot of the phenomenology that we see. The more heterochromatin that you add, and this is a very nice image from a paper that's actually not quite out yet from Emily Brown and Doris Bachtro's group, where they look at this sort of dosage series from XO to XYY, so increasing numbers of Y chromosomes. And as you increase the number of the Y chromosomes, you're seeing a suppression of the H3K9 dimethyl signal elsewhere in the euchromatin. So it's that directly, you see it's impacting the chromatin state with respect to the epigenetic modifications. All right, and we see this manifested as redder eyes in the flies with PEV. All right. So now we have a lot more information about those Y chromosomes. We know all the K-mers on them. So we can kind of play a correlation game. So what's correlated with PEV? So it turns out many of these repeats are showing fairly highly significant associations between the abundance of that particular K-mer and the degree of position effect variegation. And so now you have to sort of pay attention to the fact that, well, those K-mers were not all independent of each other. So modulo the correlation structure of the K-mers, we're seeing at least there's some relationship between a particular Y chromosome's configuration and composition of K-mers and its impact elsewhere. And this, of course, makes sense because the tendency to bind those factors that have this transacting effect is going to be sequence dependent on the Y chromosome. So we haven't tested that directly, but there are lots and lots of hypotheses that are being thrown out by these experiments. So the next thing to ask is, so here's the Y chromosome again in our Y chromosome lines, replacement lines. Does that Y chromosome impact expression elsewhere in the genome? And this is what we can tell very easily just by doing RNA-seq across those lines. And some genes show massive effects. So all we do is replace the Y chromosome, and when we do that, we're seeing particular genes that show huge differences in the relative expression from one line to another. Altogether, there were about 612 transcripts all over the genome that show a significant heterogeneity in their abundance, where it's just the Y chromosome that's changed. So you can think the Y chromosome has this massive transacting sex-specific EQTL effect, okay? So from an evolutionary biologist point of view, you think of the Y chromosome as being this dull kind of non-recombining thing. That's not right. The Y can acquire sequences that lets it reach in and modify expression throughout the genome. So the prospects for the Y to be now very specific in how it modulates the rest of the genome is absolutely there. And we're seeing evidence for it by the very exquisite fine-tuning of the different genes that are modulated from one chromosome, Y chromosome to another. Kind of makes you stop and think. It's a totally different perspective on the Y chromosome, that's for sure. There is one feature, so we did Go analysis, all that stuff. Not too much comes out of that, but this was quite clear. There's a significant clustering of the genes that show this Y modulation near euchromatin, chromatin, heterochromatin boundaries. That makes perfect sense. It's those genes that are like white modeled 4, once you sort of are in that neighborhood, those are sort of right on knife's edge. They could tip either way depending on this presence or abundance of the heterochromatin proteins modifying the chromatin state. Okay, when we try to do association between particular K-mers or transposable element abundances and specific genes, of course you get some things that come out significant. I will point out these QQ plots look beautiful along here. It took some effort to get these linear models right, but there you go. And it did require population, all those sort of cofactors. And again, many of them are because there are many of these elements that were highly population specific. And so we kind of set those aside because Beijing is weird, but Beijing also often has a quite strong population-specific modification of these transacting effects of the Y on the rest of the genome. But there are others that are quite dramatic. Which one did I want to point out? These are all different K-mers. So ACAG is another Beijing-specific ones, but some of the ones that are not so Beijing-specific are nevertheless showing quite significant correlations of specific K-mers on a specific gene's expression. It's a little bit harder to see the degree to which a particular chromatin binding protein or factor is going to be gene-specific, but it's possible, and obviously more hypotheses to test there as well. The interesting thing is going the other way, thinking what particular elements of these either K-mers or transposable elements that we've measured on the Y impact large numbers of genes elsewhere. So this is the number of genes that are significantly impacted. So this K-mer here only hits one other gene. It's quite striking. This one hits 14 genes. AAG repeats. There's lots of those in the genome. Those affect 590 genes. So that's probably a really good binder of HP1, for instance, that it would impact so many different genes. But these are quite interesting. These are those transposable elements. You know, the telomeres of Drosophila are maintained by transposable elements, head A and TART, and those impact huge numbers of genes in a transacting way, the relative abundance of those two proteins. All right, the last little bit is on the mutation process. So you think about it, well, these are sort of kind of like microsatellites. A lot of work was done on measuring microsatellite mutation rates and patterns. And this idea of slip-strand replication slippage is obviously going to be there. Any kind of long tandem array like this is going to be suffering the risk of this kind of process, and it can make these repeats expand and contract. But at what rate, and what are sort of the patterns of it? How could we tell that? And we were fortunate enough to have Julian Flynn join the lab. She was had just done an honors thesis as an undergraduate at McGill University, and it was with Daphnia pulex. It has a nice compact genome of 200 megabase pairs, amazingly short introns in Daphnia, if anybody's interested. And they also have relatively few transposable elements, a little bit less heterochromatin in their genome than many, giving them a fairly tidy, compact genome. They can be maintained strictly asexual, so for doing a mutation accumulation experiment, they're terrific. So you just maintain these sort of asexual lineages. They took one line that was very well characterized, split it into 28, kept it for 82 generations, or kept them for 82 generations. And then in a paper that was just appeared with Melanie Christensen at McGill, they published a paper on the kind of genome-wide mutation rate. So it was like, what's the estimation of mutation rate in Daphnia using this direct mutation accumulation? So I asked Julian when she arrived, do you have access to all the raw read data? And of course she did, and so we were able to run this K-mer analysis on these as well. I should point out this as well. They also ran a 15-liter aquarium, started with that same lineage, where they kept very good track of the density, the population density within that aquarium. So this is sort of a mutation accumulation experiment in a population where now selection can occur. So the idea of mutation accumulation lines is there's such a small population size, you're fixing any mutation that occurs each generation, and so you don't have any drift. It's just simply mutations accumulating. Whereas in the aquarium, you can have both drift and selection occurring. If one or another mutation is advantageous or deleterious, you can see the consequences of that by changes in frequency in that aquarium. So we'll get back to that. So they looked at these, or we looked at the K-mers within these Daphnia and had to do a GC bias correction because they didn't do a PCR-free library. It still works pretty well. This K-mer analysis works pretty well with libraries that are PCR-based. It, of course, does down rate the GC-rich repeats to some extent. Anyway, the results were there were a number of K-mers that were available to be scored across the whole set of lines. And what are we basically asking? So there's a certain abundance of a given K-mer in each line, and we're asking, does the variance in that copy number inflate over time? That's basically the question. And one other point I need to make is that the K-mers are not all independent of each other in the sense that they are connected by some degree of mutational steps. Some of those mutational steps include addition of lengths to the word. Some of them are simple substitutions, but there's unusually high degree of structure in the Daphnia K-mers, which was kind of intriguing. I think it had to do with the fact that they were asexual for so long. This is indicating the number of copies that change per copy, and this is the zero line here, which is sort of, okay, so each given line didn't change in mean up or down, whereas the bars that are going down here are K-mers that seem to decrease a little bit in abundance. The ones that went up increased in abundance. And overall, there is a tendency for the situation of these mutation accumulation lines to gain heterochromatin. Most of these repeats are gaining a little bit. So there was a bias in that direction. We could also actually directly estimate the mutation rate by this change in the copy number per generation per copy, and it tends to be in the range of 1 to 4 times 10 to the minus 3. It's a fairly tight range across the different K-mers, and whereas the microsatellite mutation rate is on the order of 1 times 10 to the minus 4, at least an order of magnitude rare. And this is in part because of the very high copy number of the heterochromatin. We think it probably makes it a bit more mutable. So going back to that cage, this is showing the coefficient of variation. That's the standard deviation over the mean for each of these different repeats. And we're comparing the MA lines, the mutation accumulation lines, against the population. For, I think it's the case, every single one of the repeats, there was a lower CV in the population in the MA lines, despite the correction for the affected population size that we did. And so what we're concluding is that there's kind of evidence here even for some degree of stabilizing selection. That expansion seems to be selected against somewhat in that population cage. And of course, we need to document that a bit better as well. Okay, so quickly, conclusions. K-mer analysis of reads provides a reasonable way to quantify the differences in these things. We saw a lot of population structure. A classifier lets us identify population of origin from nothing other than these reads. These Y chromosomes are massively variable in their heterochromatin, and the Y chromosome has this very strong transacting expression effect. The K-mers and transposable elements have a lot of correlation structure to them. We think that's probably generated by the mutation process. And the composition affects both position effect variegation and gene expression elsewhere in the genome. And finally, the whole process seems to be not strictly neutral. So the individuals that were involved, I mentioned most of them. Yassi is doing CRISPR to knock out some genes on the Y chromosome, other perturbations of the Y to see what that does. Dan is, of course, the co-PI. Sarah's looking at chlamydomonas. We have some chlammy data as well as a mutation experiment. Ian did the GC bias correction, and everybody else I've already introduced you to. So I'd be delighted to take questions. If you don't have one, quick, I'll ask one myself. Eric.
So, hi. I'm up here.
Sorry.
Here.
Yes.
Many thanks for the talk. I was just wondering, when you showed the dosage effect of the Y chromosome onto H3K9 methylation, I think there was also an effect of the X chromosome. And I guess what makes sense, but I was wondering if you also had a look into that, and if you assume that the dosage effect is in general across chromosomes, not only from the Y chromosome.
Okay, so that actually is Emily Brown's work. And if you compare XO male to XY male, they still have the same dosage of the X. And you see the phenomenon very clearly in that pairwise comparison as well. Does that answer your question?
I thought when you compared the XO to the XX, actually, that you saw some dosage effect there as well.
Yeah, no, that is true also. And that's happening for other reasons, I suppose. So, yeah, I'm not sure why.
Yeah, so...
Other reasons.
Okay. Thank you.
Thanks for a great talk and a really provocative one. So really two linked questions. One way to test directly that it's a sink for these heterochromatin proteins would be to see if you can phenocopy this effect by decreasing the expression directly of those heterochromatin proteins. So seeing, now that you've got all these different genes that respond in different ways, you can see if you can map different genes to different proteins. But if in fact that's the case, that it's acting as a sink, you would think the organism could actually just modulate the expression of those genes itself. Why go to the trouble of lugging around these big heterochromatic regions?
Oh, yeah, you're sort of asking about the adaptive reasoning for it. And I wasn't particularly making the argument that we have all this heterochromatin on the Y in order to do that. So
it could be an effect of having it.
Once it's there and you get the mutation and it's favorable, off you go. That's the sort of way evolution works. But no, this idea of specific modulation of the heterochromatin modifying proteins like the histone transacetylase, the SUVAR39. That's exactly what we're doing. And,
oh, good.
And that's very doable. So I guess that's about the end of that story. So the bigger question of, so how did all this get started? How did it evolve? How does it differ? Is we're just barely kind of scratching, like, how do you formulate that question? It's really a tough one. Okay, can I ask myself my question now? So the question is, but what about non-flies? We've heard about human and non-human. I'm going to ask about non-flies. So the non-flies, like human, does this happen in people? So what about the human Y? The human Y varies from 30 to 60 megabase. I mean, it varies by a factor 2 in size from one male to another here, mostly because of amounts of heterochromatin. And it's a difficult experiment to do in humans. It's not easy to make Y chromosome replacement men. And so you turn to mice first. And there was a very nice paper just published in PNAS earlier this year by Krementsov and colleagues. And Joan Nadeau years ago made a series of mice called consomic mice. Those had a single chromosome replaced. And one of them was the Y chromosome. So he actually made the same thing that we did with fly, the Y replacement lines. He made Y replacement mice. And there were 11 different consomic Y strains that Krementsov got his hands on, all in the same C57 black background. And this paper is a little strange because it's motivated not in terms of like this interesting Y stuff. It's just like, why are there sex differences in immune response? So this is the design that they chose to ask that question. And so they infected them with influenza A, and this is the survival curve of those 11 different lines, and they're dramatic differences. So they saw these differences and said, wait, the Y chromosome is affecting the immune response that severely? What is going on? And so they did a whole bunch of things. One of the things along the way was they said, okay, let's do RNA sequencing just to tell which of the Y-associated genes are impacting the immune response. And they found 130 genes that were affected that were on the autosomes, most of which had nothing to do with immune response. And so they thought, okay, that's not terribly informative. They did find a couple that they could sort of argue were related to the immune response. But they also then ruled out, okay, it's not SRY, it's not testosterone level, because that doesn't correlate with this. There are no other known Y-linked genes that have it. So it's just something else on the Y chromosome that's giving this bizarre effect. And that's how they left it. So I'll leave it to your imagination to consider whether it's anything. I mean, I haven't proven that it's anything other. I mean, to show that it's heterochromatin is not going to be terribly easy in this case either, but it certainly is a tantalizing possibility.
So the correlation with the amounts of Y repeat?
We don't know yet. That's the first experiment to do. Yeah. Wow, that killed the discussion. Sorry. Is Aviv here? Aviv is here. Yes.
I was wondering, is it possible that in some of these genes where you see a very strong effect, that there's actually short repeats in the genes themselves that is driving up your RNA-seq signal? So it's something that we've seen before with segmentally duplicated genes like the DUX family of genes in humans. It seems possible that if there was a short K-mer in the actual coding sequence, or perhaps even in an intron, you might see a huge upregulation that was not necessarily real.
Well, I don't quite see how that would happen. So the RNA sequencing is not done with respect to the K-mer. That's just straight plain vanilla RNA sequencing. So if there were a K-mer repeat in there, it wouldn't particularly impact that, right? Maybe I don't understand. Yeah. But certainly it is a possibility that there might be motifs that are bound by proteins that would otherwise be impacting chromatin state that could somehow affect expression. But it doesn't seem sensible that it would have that magnitude of an effect. It's really it was a huge, fourfold increase in expression. Yeah.
So the amount of heterochromatin and heterochromatin protein seems to be quite tightly stoichiometric in terms of regulating gene expression and downstream consequences. And yet, there are species like Drosophila pseudobscura where you have so many different types of Y chromosomes segregating within the species for a long time. And even in Drosophila affinis, where you sometimes don't have the Y at all. Why do you think that, on one hand, it's so sensitive to the amount of Y chromosome heterochromatin, and yet you see so much variation over long evolutionary periods of time? How does the system buffer it?
Good question. Yeah, so the response of the genome that's sort of under stress, like let's say it has an excess of heterochromatin, so the chromatin state's out of whack sort of genome-wide, it can probably respond to that. It's a very polygenic sort of trait, so it probably can respond to that by attenuating expression levels of the many heterochromatin proteins as well. But we have no idea really how it works or what's the dynamic of change of one or the other. The main thing that we did observe is that the dynamical sort of process of the K-mer repeats changing is very rapid. I mean, it's interpopulation level. In melanogaster, you're seeing quite dramatic changes already. So, yeah. Almost all these questions, you think, okay, is there an experiment that you can do? And many there really are not very good experiments to do, but some of them could be pretty incisive, I think. Yeah.
Do you have any hypothesis why, for example, Beijing population was so different in your experiments?
So this has been pointed out in the literature. There are many aspects of the Beijing population that are different. And I don't know if you can say why. I mean, it just happened that they had a trajectory where they were sort of isolated enough, marching off enough. It's possible that the Beijing population was somewhat more isolated after the flies also showed a sort of out of Africa event. Relatively little influx of European flies perhaps could have been part of it. I don't really know what the best theory is. Any fly people here who can? Yeah, I think it's population history. Yeah, that's unique. I mean, that's the thing about these heterochromatin repeats. They can just march off in some strange direction, and there's nothing particularly that's going to stop them. And if it has these transacting effects, it can have a dramatic phenotypic effect as well.
Up here. Way up in the top right here.
Wave a little bit. Okay, yeah.
Hi, have you ever seen any evidence for hybrid incompatibility or hybrid dysgenesis resulting from Y replacement in these strains that specifically you might attribute to the satellite loci rather than transposable element load?
Nitin, you'll have to help me with that. Are there specific infertilities from just the Y? So you put the simulin's Y in melanogaster will be sterile. Isn't that right? Yeah.
Right, but these, yeah, the genome, the global diversity lines,
Oh, at that level, yeah. So, so we actually have done a bunch of intercrosses between them to try to look for incompatibilities between them. But the question we were asking wasn't so much about the heterochromatin because it's unlikely to have that dramatic an effect where it would be actually infertile. What we're looking for is release of transposable elements because of loss of pi RNAs. That's a much more likely kind of incompatibility that you might see if interpopulation crosses.
Yeah, I was wondering, yeah, exactly where I was headed, whether those things are leaking into pi RNA populations.
Yeah, and the short RNAs modifying this, that's another whole story that I didn't even touch on, so leave that for later. Any more? Yeah.
Can you talk about centromeric repeats? And so in mammals, how do you expect those to contribute relative to Y chromosome repeats?
So, I mean, there are constraints on the centromeric repeats where it has to be able to recruit a kinetochore and do the whole thing of, chromosome segregation. So there's a very clear functional constraint on centromeric repeats. But apart from that, and the fact that the Y chromosome is only in males and has all those other features unless recombining it, the Y is somewhat just freer, I guess, to diverge in crazy ways. There's not much more to say than that, I don't think. Yeah. I'm eager to hear Aviv's talk. Anyway, thanks.